Now let's talk about the functional annotations information in aggregated view of proteins. Here you see a plot vista viewer, which shows you predicted and evidence-based annotations in gray and blue respectively from various PDBe-KB resources. So this annotation section has all the functional annotations coming from our different PDBe-KB partners. In order to know the details about each PDBe-KB partner, you can hover over the resource name and it will tell you the details. For example, over here you see Metal PDB is a database of metal sites and biological macromolecular structures. And similarly, you can look at the details about other resources while hovering. If you hover over a residue, you can also see the annotation details in the tooltips. For example, over here, the cysteine 1732 is coming from metal PDB, and it says that it is a metal binding site. Then you also have domain mappings, ligand binding sites and interfaces over here. And this allows you to compare all the annotations from resources from what you see in experimental data or what is seen in structures. We also have information on sequence conservation coming for calculated for the entire UniProtKB. This is how the zoomed in version of the sequence conservation view looks like. If you zoom in the details, you can order the residues based on amino acid property or probability. The multiple sequence alignment file can also be downloaded using this button. If you collapse the sequence conservation view, you can see the high level details on the conserved residue. And if you hover over it in the tooltip, you can also see the conservation score. We also have variant information in variation section, which gives you details about various sequence variants on a protein function and their relation to diseases. You can filter based on the variant effect and their origin. In this tab, you can download all these annotations which you see over here using your annotation download button in a CSV format.